mobile brain-body imaging, and the question that I have, and that's been driving me for some years now, is what kind of data, what kind of results do we actually see from these new approaches that we have that I think are necessary to drive the field? And I'm pretty happy that Stefan just dived into some aspects of the definition. I was free enough to adapt a little bit. So for me, mobile brain-body imaging and mobile EEG are methods to record, analyze, and graphically depict. So it's brain imaging method. And I started this discussion of whether EEG can be brain imaging or not, because the traditional imaging field does not consider EEG to be an imaging method. But as Stefan said, I think we have ample of spatial resolution, plus high temporal resolution. I would clearly consider EEG to be an imaging modality. So we record, analyze, and graphically depict brain dynamics in actively behaving participants. That's the important point, including ecologically valid scenarios. So we're driving natural cognition and trying to understand the neural dynamics of natural cognition. If you dissect that a little bit more into mobile brain-body imaging and EEG, I think we're talking about a continuum, obviously, from mobile EEG to mobile brain-body imaging with several dimensions that increase in complexity. For me, mobile brain-body imaging is an integrative approach to investigating brain, movement, and cognition. And this trias, all these three factors interact. If I actively behave, that will have an impact on brain dynamics as well as cognition. And so movement will change brain dynamics, but also intent, cognition, cognitive goals will change movement and brain dynamics. So it's an interaction with all three factors that we have to consider if you want to understand natural cognition. And this requires synchronization of multimodal data in relatively complex technical settings. So that would be one extreme end of this continuum from mobile EEG to mobile brain-body imaging. Mobile EEG, in contrast, if you go to the other extreme side, would be a method to investigate brain activity in ecologically highly valid scenarios, investigating the brain in the real world. And this does not consider detailed movement dynamics. Usually, movement is used to inform data analysis approach to get rid of artifacts that are due to movement, for example. But the last sentence here I just put up from Stefan's talk a few minutes ago. I think this is a nice approach.
I never said this.
This is a super nice approach really to start having, we need definitions of what these methods are to be able to clearly communicate what we are doing and why we're doing that and what we're doing. So I think for mobile EEG to have portable, motion tolerant, small and wireless, unobtrusive and comfortable systems that use additional streams, this is moving into mobile brain-body imaging. The more motion capture devices we put on the body, the more fine-grained the information about active behavior will be that informs not only artifact rejection, but also the neurodynamics associated with movement and the change in cognitive functions associated with that. So this would be my take on that. And the reasons for MOBI from my perspective are pretty clear. Stefan made some better points here. But I think if you look into the real world, we are not dealing with artificial, dark, experimental rooms with highly controlled, no head movement, no eye movements, just stare at the monitor. But we are living in a dynamic environment. We have to adapt to the activity movement of social agents. We have to adapt according to our movement goals and cognitive goals to whatever happens in the environment. And this will change drastically. This will change cognition, this will change brain dynamics. If you look at the movement itself, this is Maria. This is Maria. And this is a single cell recording. Maria is like one of the rats that won the Nobel Prize in 2015. So this is a place cell that you can hear. And obviously, this place cell is informed by active behavior of the rat. Whenever the rat is in this region of this environment, this place cell will fire. This same place cell will fire in a different region in a different environment, and there are thousands of place cells, obviously, giving feedback. And this is like a metric, allocentric feedback about the position of the animal in space, irrespective of egocentric heading, orientation, movement, and so on. And there are grid cells and tons of other cells that are driven by the active behavior of the participant, or the rat in this case. And if you even go a little bit more fundamental, so movement will inform neural dynamics, or will change neural dynamics, but only a change in the behavioral state from being seated or lying supine in a scanner to standing upright, potential to move, to interact, to use the affordance of your environment will change your brain dynamic state. If you look at Weird and Dickinson's study of Drosophila melanogaster, these are like single-cell recordings in the fruit fly and mushroom body. This is not related to direct motor control. It's like an intermediate area between visual cortex of the fruit fly and motor areas. And what you see are two conditions I'm going through. This is a fly glued to a stick, basically not able to move, resting on an air-puffed mouse control ball, basically. And we have two conditions. The one where it's like resting and not flying, and then the first, I'm going back. This is resting, not flying. And you see that there is not much activity in these areas, in the 4 neurons that are recorded here. The moment that the animal is airborne, it's not moving in space, it's just flipping its wings, right? It's still tethered, glued to the stick. The moment that the animal is airborne, neural dynamics drastically change. And this is not motor-related behavior that you see here, reflected in neuroactivity. This is an intermediate area between visual cortex and motor behavior. And it is manipulated by flying direction, for example. So these are perfect examples of a very good assumption that we should not consider our brain dynamics that we record in a lab in controlled condition as being equal to the potential of move and interact with a dynamic and complex environment. These things will likely change. Nonetheless, we still traditionally use completely stationary participants. We reduce the dimensionality of the data to then basically pinpoint millions of bits per second that we record with electrophysiological recordings, be it fMRI or EEG, MEG, whatever it takes. And then we reduce it to compare it basically to 1 bit of behavior that is the button press at the end of the trial. And this is a dimensional mismatch. And our brain arguably did not develop to have like this reductionist behavior interaction with the environment, but our brain developed to optimize our complex outcomes of behavior. And so in this sense, we're not analyzing and trying to investigate natural cognition and the neural dynamics, but we see a very important but very sparse aspect of the cognitive architecture and neural dynamics underlying that. Okay, I'm going to show a few examples from our lab on how we do mobile brain-body imaging, down to how we do mobile EEG. And the main idea is that I'm going to dive a little bit into the results of temporal domain parameters, eventuate potentials mainly, that we investigate on the sensor level, but using different approaches, mainly ICA to get rid of artifacts, to more frequency domain-based analysis approaches with longer time periods of analysis of the data, to come to the end, basically, or in the end, I'm asking the question, how can we accelerate the field? How can we systemize our approaches? And the main question that we should keep in mind here is, what are the parameters that we record? And are these comparable to the parameters that we know that have been established over the last decades using traditional EEG, MEG methods? And I think a few of these aspects will be replicable and highly reliable. But I also assume that some of them will not. And the question is, how do we approach it? So basically, how to image or how to run mobile brain-body imaging approach. This is a lab that you can see here, the Berlin lab in the background. So it's like a 150 square meter lab space dedicated only to motion and synchronized brain motion recording, so mobile brain-body imaging basically. And what we want to do is, like, if we want to have like brain dynamics, investigate that, we obviously need some device to measure brain activity. That can be FNIRS, can be EEG, and these are the two modalities that are currently accessible for mobile recordings. In the future, there might be MEG coming up, some recent developments that are really exciting, but not yet far from mobile. And if we want to understand how active behavior influences brain dynamics, then we have to synchronize that with behavior. So we have to get behavioral recordings, motion capture in this case. So we synchronize brain dynamics with motion capture. And I think a third pretty important aspect of mobile brain-body imaging and also mobile EEG is the experimental protocol. And how much do we restrict interaction with the natural environment by using standardized protocols?
and how does that have mediating between established parameters that we know from the last decades to what we will get with mobile recorders. And this is usually what we do in the lab mostly is head-mounted virtual reality. And all these data streams have to be synchronized, and you all know that. Most likely we're using the lab streaming layer architecture for that. And this gives you amazing options to also manipulate your experimental protocol, right? So you can use position, you can use movement, you can use brain activity to inform your protocol and change the protocol in near real time. So this is a very powerful tool. And as Stefan said, I think we still struggle with like jitter in the system. We still struggle every single time new hardware components have to be integrated. And one of the aspects that we should, well, basically establish in the field is that we have like all possible hardware configuration being tested for latency and jitter. And this should be, put out there somewhere publicly for everyone to assess. So we started doing that in Berlin, and I invite all of you to, do the same, so that we can get this data, this kind of information for future recordings. Besides LSL, which would be the synchronization of multimodal data during recording, obviously this is not the only problem, but then you have to analyze multimodal data. This is just another problem, because we simply have no idea of how movement informs brain dynamics. So if I want to grasp my bottle of water, is it the endpoint of that grasping movement that is controlled by the brain? Is it like, what aspect of the movement itself is controlled by the brain? We have no good idea about that. And obviously, we have to explore a lot in the data, but basically, irrespective of how you address the data, you have to have some kind of multimodal common analysis of this data set. And this is non-trivial at all. What you see here is the Mobi plugin that is not well developed yet. Alejandro Ojeda started developing that. It's not a standardized tool in the EEG lab. It's open source, basically all know EEG lab, I assume. So this would be one approach to analyze the data, but obviously different labs develop different pipelines, and there's a plethora of different pipelines out there, including every single lab's using a different pipeline to a certain degree, completely justified, because every data set you record, depending on the environment, the protocol will be different. So there has to be flexibility in this address. The question is, can we burn down to the very common aspects that are the same across different experimental situations that can be established a pipeline to address crap like that. So this is basically data that would look like, I think, one of the very first experiments that we did at the Swartz Center. This was and with that I'm coming to the first temporal domain or time domain parameter extraction in mobile EEG recordings. A treadmill study where we investigated standing, slow walking, fast walking, and running participants that were working on a visual secondary oddball task. To get back to the point, you see a picture in a second. So this is the data. This is seconds on the time scale that you see here. This is the 256 channel system that biosemi that we recorded at that time. And you see like the scale here, like that somehow is like 50 microvolts. So the artifacts that we have here in the data are massive. And basically, if I had recorded that in 2000 to 2007, I would have said, okay, let's get another subject. But we were able to analyze that, and one aspect of that, and Arno might talk a little bit about that, is ICA, independent component analysis, to decompose basically brain from non-brain activity. And I would not call non-brain activity per se as artifactual activity. Muscle activity, eye movement activity are components that serve a cognitive goal, right? If I move in space, if I walk on the treadmill, my head is displaced in 3 dimensions. I'm going up and down, I'm hopping back and forth, I'm going to the left and to the right. And this is going to be compensated by at least, well, we have like 2 times 12 muscles only in the dorsal neck, plus eye movement. So if I do, and I see like this black and white striped thing flying towards me, I want to be sure that I have that in focus. So I have to compensate my head position in space, and the rest will be compensated by eye movement. Actually, the biggest part will be eye movement compensation and then neck muscle. But if that was basically on the bee is going to approach you, you want to see that where it is. So this is part of reaching your cognitive goal, and it's informative. I will come back to that later. So If we want to dissociate that from mechanical artifacts or other kinds of artifacts, electrical artifacts, electronic artifacts from the gear that we wear, then ICA is one good approach, but definitely not the only approach to analyze the data. So what you see here is a simulation of Zeynep Al-Achar from the Swartz Center. She simulated like two alpha sources in the parietal cortex and the motor cortex on the left hemisphere, parietal cortex in the right hemisphere. It slowed down, it's like 9 and 11 hertz if I remember correctly. So the movie is slowed down, and then you see like the back projection to the skull on the right side here. And what you see, like as you all basically know, you basically get a linear mixture of all sources being active, what Stefan just referred to. If you stick several electrodes in the ear or around the ear, you will get activity from all sources in the brain being active at the same time. That's one point, and then obviously you get that also in frontal cortex, and not only directly related to the areas where that the signal originates from. So this is the study that we were talking about, that I was talking about. So this is a subject running on the treadmill. We had multimodal data. This is the visual outbound task. Participants had to respond to the X. The standards were a plus sign. And we recorded multi-channel EEG, 256 biosemi. We had EMG activity from the legs. We had a dual force measuring platform under the treadmill. And then we had like motion capture on top of that. So, and we used basically the signal to get information, to use movement data, the gait cycle, to extract artifacts from the data set that I just showed you. And we were able to dissociate running related and walking related artifacts from brain dynamics, and could analyze the P300 to this target even while participants were running. And that was based on the data I showed you. Going further down to mobile EEG, then obviously you reduce, my perspective, the number of technical components that you record concurrently to gain more movement or freedom of movement, flexibility to interact with your environment. Because if you look at that, it's not really natural movement, right? 256 channels on your head restrict natural head movements. A backpack with like 2 times 3 kilogram amplifier restrict your natural. So this is not completely natural. And if you add motion capture, like several sensors to that, then it's not going to be natural. This is natural, more or less. You still have a backpack, but you can move free. You can drive your bicycle, you can sit outside, do your task, you have like all the things that Stefan was talking about. And you gain considerable freedom, even with systems that are unobtrusive, near invisible. And I am pretty sure that this is the future, really, if you talk about applied EEG in the field. For me, going back to basic research, this is not sufficient, however, if I want to understand how the brain controls behavior and how behavior influences cognition and brain dynamics. A combination of both would be superb. So if you can get me some of those, I'd be okay. But so our direction basically in Berlin will be to try to understand the very basic influences of behavior on cognition and brain dynamics. If you look at the mobility, obtrusivity, and possible analysis approaches, I think there's like a more or less linear relationship with increasing channel density on the abscesses that reduces your mobility but increases the potential range of analysis approaches to the data. If you have only 2 times 8 channels, then obviously possible analysis approaches are reduced to a certain degree. If you do have 256 channels, you can do lots of analysis approaches, but you're not as mobile as you would like to be. And this is the last slide on that theoretical background. So there is a limited approach of data analysis for high mobile experiments, and more extended analysis options if you go with higher channels. Okay, so I think the question really is what do you want to do? And obviously, if you're interested in real natural behavior, and you do have specific features that you're interested in, and I think that's a good point. If the only thing that you need to know is onset of epileptic seizure, for example, and whether it is timing information, for example, then in-ear would be sufficient. If you're interested in a P3, and Stefan and his lab showed that, that you reliably classify P3 in an oddball task outside, then if that is a feature you're interested in, why would you need more? If you're interested in more, in a higher complex combination of behavior and brain dynamics, then you need more channels. Okay, so, and now I'm coming to the examples. I'm starting like with 3 temporal domain or time domain parameter experiments that we focused on, and then switch to a more complex frequency domain experiment. And the very first, basically, experiment that we did was this kind of cognitive mode interference in dual task walking. This is the treadmill study that I was talking about. Stephen was talking about cognitive mode interference if you move outside into the world. So if I look at the restricted Oldenburg campus environment, there were not much activity going on. No social agents, no cars. No, like if you're in Berlin, this is different. You're afraid on the street.
We have a few social agents in
So dynamics in the environment will have an impact. I'm coming back to that topic. But motor activity will also have an impact. Julius Reiser from Edmund Wascher's lab in Dortmund just came out in scientific reports showing different motor complexity to have an impact on these established time domain parameters. So we thought, like with this very first experiment, okay, let's do like a comparison, and we simply assumed that standing would be easier than slow walking, would be easier than fast walking, would be easier than running. And in all these conditions, participants have this secondary oddball task, a visual oddball task. And what we did is we used ICA. So this is the setup on the left-hand side, and you see all the sensors that we use for the setup. And we ran ICA, color-coded here. You see like in yellow IC clusters. So we use k-means clustering on standard ICA decomposition. In yellow, you could see clusters that we associated with neck muscle activity. They are actually nicely aligned with the skull. If you look at this alignment, this is, I think, very interesting to see. And this was the infracerebral cap that we used. So it's like kind of deep. And most of these neck muscles attach between the inferior and superior neutral line on your skull. So there's a very common placement of most of these outer layer muscles. And that seemed to be straightforward to localize even with a head monitor that does not exclude one single muscle. And then color-coded here, you see brain components, and in gray you see eye movement components, or reflecting eye movements. Or clusters, and these are the scalp maps only of the brain components, or clusters of brain components back projected then for the further analysis. So, and one of the nice ways to visualize data and the contribution of different brain regions to the surface parameter that you're looking at is like this data envelope that you can run. So you take all your multi-channel data, take the maximum and the minimum at every single sample point, and then you compute or visualize basically the percentage of contribution of different regions, be it artifacts, components, brain components, whatever you like. And this is what I do here. I'm going to show you the target onset at time point 0. This is the event-related potential envelope that you see. And in yellow and purple and in gray, you see muscle, eye, and brain. And it's only the ACC cluster that I'm plotting. So what you can see why people are standing, there is neck muscle activity. There is always some kind of neck muscle activity going on. Then you do have eye movement activity. This is blinks that I'm showing here, and they are, if you do not control for blinks, they will be prominent in the data, obviously. And there's also some brain activity. So this is onset of the target during standing. If you now look at the same components, these 3 clusters and how they contribute to the same envelope during slow walking, what you can see is that if you stop walking, obviously there's an increased neck muscle activity visible. I'm only plotting one neck muscle here. There's an increased eye movement activity present in the data. And if I go back and you compare the data in ACC, this is pretty comparable. The same holds true for fast walking. The ACC data like literally does not change, really, but eye movement and neck muscle activity increase. If you look at running, this would be a different pre-processing pipeline because we had to get rid of gait-related artifacts. I'm not going to show that here, even though it would be possible. It would be a different solution though. So the point is, different movements will come with different amounts of artifactual activity if you consider muscle and eye movement traditionally as an artifact. Nonetheless, we basically showed, or we were able to show, and I'm sorry that the slide's not here, this is at me, that if you compare the targets versus the standards across the 3 walking conditions, that you can extract brain dynamics associated with this kind of task during different movement conditions. You find significantly increased P300 for target onset as compared to standards, and you can do that reliably across all movement conditions. So then a second experiment going into the same direction, looking into, again, the question of cognitive state, behavioral state, and brain dynamics. So in the running condition, we just use a joystick, a remote control to respond to your target. But this, again, is like not what happens in the real world, naturally. So we thought, okay, we stick with a traditional or a well-established paradigm, a P300 oddball paradigm. So you take a visual oddball instead of an auditory oddball, even though the auditory oddball is way more often used in the field. And so usually what you have is, in this case, you have like a standard color here, black, and that changes like on 30% of the trials to a target color, like in this case red, and then you have to respond to it. Okay, that can be either centrally, can be peripheral, whatever you use. And this would be the traditional setup. You would have to stare at the fixation cross, you're not allowed to move your eyes, you're not allowed to move at all. So, and we thought, okay, put that into the real world, and we projected that on a huge projection screen. And we thought, okay, real world is also, you're not stationary, you're not restricting your focus on one aspect of the field, but you're looking around and you're moving your eyes, so we got rid of that fixation cross, and then we thought, okay, real world is complex, it's dynamically changing, so why don't we have the target move around? So this is what we did. So this is the experiment, and so target color here was blue in this block, and you see the participant basically interacting. There were 2 conditions. The one condition was that participants were standing in front of the screen and they had to intercept the trajectory of the moving sphere whenever it changed to its target color. In the second condition, they simply had to press again a remote control. So this was mimicking the traditional setup where you have like 1 bit of information after each try, pressing a button. What you see here, if you look at the response times, these are subjects on the x-axis, and then you have like a response time in seconds. For red, the physical point condition, and in blue, the button press condition. And what you see is there's like, obviously, kind of definition here. What is response time when you start moving? What we did was like taking 30% of the maximum velocity value into the z direction towards the screen. And this is a significant effect in response times, but this effect, the difference in response time, remains significant if you go up to 60% of the maximum velocity, so far into the movement itself. So what you see is physical pointing in red is significantly faster. It's a main effect across all participants than a simple button press. Exit interviews told us participants said like that the button press was really boring. That this following the pointing task was way more exciting. They had fun actually doing the experiment. It's kind of a little bit of like whack-a-mole, right? So this is like another thing. If you start gamifying your experiments, you keep up motivation into these situations, then we might change the parameters that we extract already. So, and then if you look at a electrode closely related to PZ, so this is not a standard montage, so but that was the electrode that was closest to the traditional 10% PZ electrode. You see the target onset for in blue the targets, and then you see the 2 conditions. The solid line is the pointing condition, and the dotted line is the button click condition. We had also a distractor in red that was visually distinct but required no response. And then we had like in black the standard visual stimuli. And what you see is like, of course, we do have a significantly increased P3 or late positive complex, I would call that, that differed from the standards, and also the distractor differed. But what you see here, the main effect obviously is something of a huge difference between the pointing condition and the button press condition for the times. If you look at the timing, this is somewhere around shortly before 400 milliseconds, 370 something. I just go back to the response times. If you look at that, there's obviously a potential confound here. If you simply look at the back projected sensor level activity of the P3. This could be simply a huge artifact due to this kind of rapid movement that would get into all of the posterior channels, maybe all channels. So if you only look at sensor level data, standard cleaning procedure, there might be a confound. So again, you just ran the standard analysis pipeline that we use in Berlin most of the time, did an ICA decomposition and subsequent equivalent dipole reconstruction. Again, in gray now you see the neck muscle. Again, it's like the same picture, completely different set of participants, different experiments, similar picture of location of clusters, color-coded, the brain components, and then white in the front, the eye movement clusters. Okay, and what we did is looking at parietal clusters and how they contribute to the P3 complex, or the late positive complex that we were interested in. So we picked only these two clusters, and then we back projected that again to the envelope that I showed you in the beginning, and we compared the key press and the pointing condition for the standard stimuli and the target stimuli. So what you see here is the standard. In the key press condition, you see the envelope. There is a little bit of activity, but there is no significant late positive complex. And in red, you see the back projection of the two parietal clusters. Importantly, the envelope here is based only on brain clusters back projected to the center. Okay, we got rid of all the muscle, eye movement, and anything else. It's only brain components. If you compare that to the pointing condition, there is already some kind of a difference. Gray is the analysis time window that we use for the P3, or late positive complex. And again, the posterior parietal cortex shows a little bit of activity, but it's not that much pronounced. If you look at the scale, it's like 2 microvolts. That's not the P or the late positive complex we're talking about. If you look at the targets, there is a late positive complex and increased activity in parietal cortex. And the important question now is, could we get rid of potential movement-related artifacts? Do we still see, with this kind of approach, a difference in the active pointing condition as compared to a traditional button press condition? And the answer is clearly yes. There's an increase on amplitude by a factor of 2, at least. Some participant was really pronounced. And you see a clear late positive complex. Timing is pretty much the same as it is in the traditional button press condition, but the amplitude is way enhanced. And obviously, parietal cortex would be one of the multimodal areas that would have to process different incoming sensory information about movement direction, color changes, but at the same time has to prepare a motor response. It's not only that you have to prepare a motor response, but you have to basically predict the position of the sphere, the trajectory in the future. So you have to constantly compute, online compute the position to be able to interact with that. And this is simply an increased computational demand in a more natural dynamic environment. And this is one of the points I would argue that will change brain dynamics, instead of sitting in front of a computer screen and pressing a button. So this is, for me, a simple form of embodiment, that a motor goal or the motor task itself increases in complexity and will change the brain dynamic response to a change in the environment. And now we basically use the same time domain aspects to go into the wild, really, Berlin. And walk through the city, do a navigation task. We have been studying some kind of navigation systems with the question, so the standard realm of you use a Google Map assistant and you just stay stupid, right? So me, I'm living in Berlin for 7 years now, I have no idea about this thing. Anytime I go out, I just Google where I have to go, I just follow the route. And next time in the same area, I still don't know where I am. So this is called de-skilling, right? You over-rely on an automation system that takes over the very cognitive tasks that you're supposed to do. So you're not processing information anymore, and you do not learn spatially. So we were thinking about changing that and create like the eierlegende Wollmilchsau, you all know that term, I'm sure. So like have a solution that does everything, that provides you wave following without increasing mental load, but at the same time fosters spatial learning. And we simply try to do that by augmenting landmarks in the environment. Whenever there's a decision-relevant intersection, we pick up one of the environmental features and give additional information to them. So we did that in car simulation studies, and replicated an enhanced recognition effect for navigation-relevant landmarks. And the question was, at one point, can we see that also in a realistic environment? This is Berlin. We had like a route through Wilmersdorf, and you see like people, this is the standard environment in Berlin. Nobody cares if you're running around with a 60 This is by far not the most exotic thing you can see in the city. So that was nice to see. You can leave the campus, you can go into the city, nobody cares. The question rather is, how do you get relevant information out of the data? How do you get to event-related information? And so we basically picked up something that Edmund Wascher did. This is a route through Berlin. This is a suburb of Berlin, and we basically depicted navigation-irrelevant landmarks that were alongside the route without navigation decision, and navigation-relevant landmarks where you had a direction change, and then we showed information only for the relevant landmarks. The experiment was like about 1 hour plus preparation time. We had to drive participants to the starting location and things, and after that we got them to the lab to do a recognition test. This is a scenario, and we used blink-based activity. So being forced to come up with events that are not present and not standardized without a scene camera and some kind of machine learning approach to get standardized events from that information stream. We resorted to a blink-based approach that Edmund recently showed, Edmund Wascher, in a work scenario. And if you look at the number of blinks, these are significantly higher than they should be if they served only moisturing function for your eye. Right? So every time you blink, your eyes get moisture. But if you want to do that purely for moisturing function, I think 1 blink per every 30 seconds or so would be sufficient. In addition, blinks systematically covary with fatigue and mental load that was shown several times. Blinks allow for non-obtrusive recordings because people simply don't realize that they do it. It's not a conscious thing they do. And they are related to visual processing. So there are several studies in this area that show that blinks are relevant to information processing, and I have to run. So what we show, just jumping through some slides, standard navigation instructions decrease recognition rates for navigation relevant landmarks. Enhanced instructions basically increase recognition. And this actually is an effect if you compare that to the irrelevant landmarks that seem to indicate, and we have that in several experiments, that if you use standard instructions, you suppress spatial learning actively. And what we do is we eliminate this effect by using additional information. If you look at the blink-based potentials, they are really neat, nice, clear. And here I'm coming to the very first problem that I have. I have no idea what they stand for. Okay, what we see is a significant difference in the instruction conditions, but this is not your standard P1 component. And what does it mean if you get these blink-related potentials? And moving into a more complex setup, this is heading computation. Now I'm walking from the temporal domain to, I'm threatened already, please take the clock away, to the frequency domain. I'm running through that, I'm late already. So we did a simple heading computation task. And people had to rotate following a visual dot, basically, that sat on and rotated unpredictably between 10 to 180 degrees. They had to follow the dot, change the color, and then they had to move back and indicate their original heading. And we did that in 2 conditions. Again, we always compare that, right, between standard lab setups, traditional, and the new one. So in the standard replication, the same visual input was controlled by a joystick. In the MOBI setup, you controlled that by active rotation. And we show significant differences in behavior, with mobile rotation being way more accurate because you have vestibular information, you have proprioception, you have everything that appears naturally. And we reconstructed some nice clusters, and the one you see here in red, in the retrosplenic cortex, which is my favorite brain region, because it's kind of translation hub between sensory egocentric information into allocentric information that is stored in the temporal, and vice versa. That's simply a different way to show that. If you look at the behavioral states, there are differences already in the baseline before you start rotating that indicate that the future upcoming motor task might change your brain dynamics already, even though you're not yet behaving. And if you look at the activity pattern and the difference between what you see here is a baseline corrected Hilbert transform in power in different frequency bands for different velocity bands. So we simply chopped the data dependent on the velocity and sorted them relatively from 0 to the 100% fastest trials. And then we investigated the spectral power for these segments and contrasted that between the rotating and the joystick condition. And we show that there are very specific frequencies indicating a significantly enhanced signal in the actively rotating task. I'm not going into the details here. This is a very, well, it's still not mechanistic. It's not mechanistic, and this is like, we can show there's a causal relation between brain dynamics and behavior. And we have to get to that point. That's like another issue that we have. If you look at the retrosplenic cortex, this is a joystick condition. This is like your traditional alpha band that shows the classical desynchronization that we always see. This is your active rotation condition. And it's nothing at all anymore like a standard traditional setup. Okay, this is a difference, it's highly significant across a wide band of frequencies. And this is obvious, we can discuss that later, but if you look at single neuron, second order vestibular neurons in monkeys during active head rotation are silent as compared to the same velocity rotation when the monkey is passively rotating. This is predictive processing, this is reafference principle. I know what kind of movement I'm going to execute, I know what kind of sensory information is coming back from the movement execution, and proprioceptive neurons cancel out vestibular neuron activity, second order level. So there's no activity because I know I'm going to get this information if I do this. The only residual is visual change. This is what I have to focus on. So, and there will be drastic differences in these kind of setups. This is like the question. So if you run, if you move, this will change sensory input to the system. At the same time, this sensory input change will have to be processed in the brain, leading to a change in brain dynamics. Then, in addition to that, there will be environmental complexity that might be hardly controlled at all. That will change sensory information processing. As Stefan referred to, if there's like a Berlin angry bicycle approaching you, there's attention that you have to withdraw from your primary task. You're not staring at your mobile phone anymore because you don't want to collide with that angry bicycle driver. So that will change how you process information. And we have to address that systematically, and we have to see how all these different factors are related. So, temporal domain, to make the point short, I think there's a huge overlap with what we can see in traditional parameters, features. And this is by far not what happened during the last years. There are more studies, but most of them point into a similar direction with respect to, for example, secondary task, motor cognitive interference, or P3 oddball. Most of them are auditory, by the way. But temporal domain parameters might be replicable. There will be degrees of variation depending on the noise level that also interact with motor complexity, changes in motor task, and complexity of the environment. However, for frequency domain parameters, I would say that this might be difficult, at least if you're talking about event-related frequency changes and not induced frequency changes. So we have a number of aspects that will have to be considered, and I think that the field will benefit from a systematic approach of replicating this. And this is my cry out to the community, basically, that we should kind of organize ourselves and try to develop a framework how to address this issue, not to simply produce information, but to come up with knowledge model-based. This is something that's not really happening and hasn't been happening in neuroscience, I think, for the last years. We have amazing opportunities, methods to work with that, but we're not producing knowledge. Okay, with that, I'm like,
Thanks for this really, really interesting talk. But you're producing information at such pace that you don't know what to do with it, with all your sensors and high density, you know.
I totally agree. So
you're part of the problem.
Totally. I didn't say I produce knowledge. I don't, unfortunately. But this is like one of the aspects that all these data sets should also be publicly available, because like one PhD student can work on one of these experiments for the next 5 years. The dimensionality is we have to share. We have to have like different labs approaching the same data with different perspectives.
Yes, I think everyone who has attempted to make even a small lab can only imagine how, just like what effort it takes just to make these things work together, let alone do the further scientific things. So I think we have time for 1 or 2 questions.
Thank you, Klaus. I was interested in the experiment of the wayfinding in Berlin, especially when you said that you were using link rate to associate different places. You had that map to locations, right? And when you had an increase in link rate, you found that they were in a very highly recognizable location. Was it?
The data actually, so there was no significant difference if you look of the into the overall difference in weights. So this, there's no significant difference in the number of things as a discussion conditions, which for me is good. We do have different issues with this study, so it's really difficult but this is not significant, and this allows us a direct comparison of the link rate potential, right? So if there were differences in link rate already, then any difference in the P might be contributed by the different number. But we are now trying to dissect the data a little bit more during and after the instruction phase itself, and it's not straightforward.
Because you know that in our field, the link rate is associated with the workload.
Yes.
And you know, there are a number of studies that associate the reduced link rate to higher workload.
Yes, which is cool, because our primary goal was to develop navigation assistance that does not increase mental load. So we want to have the primary task of the automation being kept. So we want to help people to navigate, but at the same time we want to increase spatial learning by the user system. So no mental load increase is perfect, no change in link rate is perfect. Still a change in event-related potentials is great.
One more? First. Let's come first.
Problem in real life situations. So if we want to, we have to assume we can have a method for us to cleanly remove all the very complex factor generated artifacts. And this has to assume that ICA will, for example, will work very perfectly in both lab setting and also in the real life situation. Is this possible, for example, in real life situation, somehow loss of artifacts actually all jump into the data, then actually ICA also works slightly differently? And in that case, the cleaned neural data that are derived by this after ICA cleaning will be also slightly different from the lab setting. So is there any way to validate this, for example, by simulating the data, after ICA decomposition, you again put the data into the brain and then see ICA can work very well in the simulation data to validate the difference between lab and real life is real, yeah.
I think that's a very good question. I don't know of any systematic study that has shown that there is
low computational demands that can run ICA on the fly online, basically, and improve constantly, adaptively. But that doesn't answer your question. I think one approach might be to have a strict comparison of the same task in the lab and outside, a very well-controlled experiment with like an over-sampling, basically spatial over-sampling. So my hope would be, considering ICA, that 256 channels is an overkill and have sufficient dimensionality to get all of the dimensions that come into the data. If you then subsample and run the ICA on lower spatial densities to see what changes, that might give you a first insight. And then the comparison, obviously, between lab and real world. That might be one approach, I don't know. But I would also clearly say that there might be more efficient and better algorithms than ICA in the future, and already out there, like sparse Bayesian approaches that use like libraries of artifacts that we can establish over time to improve source reconstruction and subsequent temporal domain frequency domain analysis. So I think there's different ones to look at.
Okay, I think you'll have also the chance to hear more of Klaus at the panel discussion and possibly ask more questions. Let's once again thank him for a wonderful talk.